How does gut microbiome impact cancer screening outcomes? Have you ever wondered how the tiny organisms living in your gut can influence something as significant as cancer screening outcomes? The gut microbiome, which consists of various bacteria, viruses, and other microorganisms, plays a crucial role in our overall health, particularly in colon health. Understanding its impact on cancer screening is an exciting area of research that is revealing important insights. First, let's talk about what the gut microbiome does. It helps digest food, supports the immune system, and protects against harmful pathogens. A balanced microbiome is linked to better digestive health and a lower risk of chronic diseases, including colon cancer. However, when the microbiome is disrupted, a condition known as dysbiosis can occur. This disruption can lead to inflammation and changes in the gut lining, which may increase the risk of developing cancer. Research indicates that certain types of gut bacteria are more prevalent in individuals with colon cancer compared to those who are healthy. These bacteria can promote inflammation, creating an environment that supports tumor growth. Some bacteria produce substances that may damage the DNA of colon cells, increasing the likelihood of mutations that can lead to cancer. Additionally, certain bacteria may affect how the immune system responds to early cancer cells, potentially allowing tumors to grow without detection. Now, how does this relate to cancer screening? Cancer screening aims to detect cancer early when it is most treatable. The gut microbiome can influence screening outcomes in several ways. For one, scientists are investigating whether specific patterns of gut bacteria could serve as biomarkers for colon cancer. If certain bacteria are consistently found in people with early-stage cancer, they could help identify at-risk individuals before symptoms appear. Moreover, the microbiome affects inflammation levels in the colon. Chronic inflammation is a known risk factor for colon cancer. If screening tests, such as stool tests or colonoscopies, are performed on individuals with high inflammation, the results might be harder to interpret, or the cancer risk may be elevated. While this is more relevant to treatment than screening, there is evidence suggesting that the microbiome can influence how well the body responds to cancer therapies. This could impact long-term outcomes after cancer is detected through screening. Looking ahead, as researchers learn more about the microbiome, there may be opportunities to personalize cancer screening recommendations based on an individual's gut bacteria profile. Maintaining a healthy microbiome through a balanced diet rich in fiber, fruits, and vegetables, while low in processed foods, can support colon health. This might improve the effectiveness of cancer screening by reducing inflammation and cancer risk. Although the connection between the gut microbiome and cancer screening outcomes is promising, more studies are needed to confirm how these findings can be applied in clinical practice. Current screening guidelines from major health authorities do not yet include microbiome analysis as a standard part of colon cancer screening. It is important to remember that while the gut microbiome is an exciting area of research, it is only one factor among many that influence colon cancer risk and screening outcomes. Established screening methods, such as colonoscopy and stool-based tests, remains the gold standard for early detection. Maintaining a healthy lifestyle and following recommended screening schedules are the best ways to protect colon health. Always consult a healthcare professional for personalized advice about colon cancer screening and gut health. Ongoing research may lead to new recommendations in the future, but for now, the focus should remain on proven screening methods and healthy living.